Welcome, Koos, and thank you very much that you are talking to an audience interested in host-microbe interactions and to people interested in our research center, Origin and Function of Metaorganisms. You are Professor of Biology at the University of Copenhagen. You're an eminent scholar in the field of evolutionary biology. You are director of a Center of Social Evolution in Copenhagen. And one of the world leading experts in social behavior in ants. Maybe the first question to you, how did you find this question? What is so burning in you now? What we think in the WiKo keeps you up at night? This symbiotic interactions, how did you got to that at the very beginning?
Well, thank you, Thomas, for having me and giving me the opportunity to tell a bit about how all this happened over rather many years now. I'd like to start saying that it was only very gradually I became interested in symbiosis. My prime interest, as you mentioned already, is really to understand ants. And after the ants, also to see the parallels in other advanced social insect social systems. Now, why ants interested me in the first place was not about the interactions they have with microbes or with any other type of organisms, but basically I was interested in their family life. So in some ways you can say that's also a type of meta-organism. It's just like members of your own species. But as most people would know, the ants are really fascinating in terms of the division of labor that they realize in what is a family, really. So they have queens that do all the egg laying. They have specialized worker castes, sometimes soldier caste. And what intrigued me from the days I was a PhD student and started to discover ants was, how did that evolve? How did it not only become stable in the course of evolution, but radiated into about 15,000 species of ants that populate the entire planet, almost. And the secret, one of the secrets of that, I realized, was something that very few people actually, I think, are aware of. That what an ant queen does when she founds a family is mate with a male, but rather than keep doing that through her life, she stores the sperm for the rest of her life. She will never mate again. And she may live for 20, sometimes 30 years. So the first really cool question that came to my mind is, how is it possible that ants have evolved sperm banks at ambient temperature where we need liquid nitrogen to do that? And how is it possible? Because they never get new sperm. They need to build these huge families, sometimes in the millions. How is it possible natural selection could produce that? And how is that distributed?
What is the answer?
The answer is we still don't know, but we become a lot closer. So I started to map, basically using genetic markers to see, are they always mating with a single male? Is it sometimes more males? So what was the ancestral condition? What came later? And this is one of the things I'm very sort of pleased about in retrospect, that I think I solved in the end by showing that a form of lifetime monogamy is really the ancestral state of the
a lifetime
monogamy. So that's how these specialized families with division of labor that is so impressive that it almost looked like the specialization of the somatic cells in my body that serve my germline. So seeing the analogy between how the termites, how the ants, how the honeybees came into being by this ancestral state, it's comparable to a zygote that founds the body of animals or plants. Here it is the lifetime committed pair that basically has no promiscuity later in life. it's the full SIP family that got established. And only later on, as a secondary development, that could be diluted somehow as an elaboration. That's why honeybees, queens mate with 20 drones, because that made sense as a later development, but not in the origin. So my initial meta-organisms were families, with all the specialization, all the division of labor that somehow became part of that, and how that unfolded over evolutionary time in different groups. And I think I found the key answer to that in my early career. So it was only after that, and after I'd settled, as a coincidence almost, not quite, but I figured to study this, I needed an ant system that has the variation in mating systems that can help me understand that. And I ended up studying the Latin American fungus-growing ants. And initially, that whole symbiosis with a fungus garden that is so special for these ants was a bit of a secondary interest.
I see.
But of course, in the course of time, after you solve the family bit of that story, it became really intriguing. So what about a commitment between an ant family and the symbionts that it has? So initially, those were fungus gardens, where we found out that that's a really strong commitment too. It's similarly for life. So what an ant colony has is not just a whole bunch of different fungi that grow in the colony. Now, it's a single clone. It is also a single clone that's irreplaceable. So once you get that established very early on in colony life, it is as tight a bond as mating with that one or several males early in life for a queen. You basically, you stick to that single clonal symbiont for life. And that colony, most of them will die, of course, at a very early age, but the ones that do survive may last for 20 years again. And that symbiont with them, it's never replaced. It's as tight
and
symbiosis.
as stable as the family also is.
Yes. So it became a meta-family with another layer.
I see.
And then we started to look at bacteria gradually.
Okay, wow.
And trying to see, do we have similar alliances that last, or is that more flexible? So cuticular bacteria produce antibiotics that help the ants to fight a single fungal disease that bothers them by infecting their gardens. And it's not a monoculture, but it is a single pseudonocardia bacteria that basically does that. There is more diversity in the population, but again, every colony has only one dominant symbiont that does the job for the ants, and which we now have the molecular data that show that they make a huge amount of antibacterials and specific antifungals. So we're starting to understand the molecular way in which this symbiosis works. In other words, the services that are offered by these bacteria to basically keep being a reliable disease-fighting symbiont. And one thing that strikes you is that this has been around for millions of years. This has been around for millions of years, and where humans all the time run into resistance problems when they start applying antibiotics in their agricultural systems, the ants have apparently escaped that problem. And I think one of the secrets is that they don't apply it very proactively, but they are very careful that they only use their antibiotics when there is a real problem. Plus that the way they grow their symbionts basically means that natural selection will keep improving the system as soon as resistance problems might arise. And again, it's one of those sort of really intriguing thoughts that the ants may have done better
Sure.
in human societies.
So many lessons you can learn, obviously, from the ants. I think it's wonderful how you, if you look back on your discoveries, and you have clearly now lined out the major discoveries which you made in your career. Actually, we came across your and Höll-Dobler's research, of course, when we were looking, we and a few colleagues in the field, on a term for our new awareness of the complexity of multicellular life as an association of many. And so we were looking for a term. And of course, it's a superorganism, but the superorganism is taken by you and your colleagues to describe the family which you are now exploring in so depth. So that's why superorganism was not what we could take. So we had to go to meta-organism, which some people don't like too much, but I think it describes what we really want to do. So now, you haven't told us who introduced you to ants. Yourself, in your parents' garden?
I think if I should describe myself, I'm kind of a natural historian who's gone molecular over his career. This is an interest from childhood. I learned it from my father, who was a school teacher. But I didn't academically grow up in a social insect group. I basically graduated in a department where most people were doing community ecology in a much more general way, which is how I started myself. And it happened to end up with ants through an essay I wrote for my graduation, through field work that I started to embark on. And at some point I was just very fortunate that my major advisor at that time suggested, maybe you work on ants for your PhD. I don't care so much about what you do, as long as it is an interesting project. And maybe this was very typical for, we're talking about the 70s now, the previous century, that academia wasn't quite so organized as it is today. So there was much more freedom to just let things happen and see whether anything interesting
and let young people do what they really want to do.
Yes.
Yeah.
And again, I mean, I would have had the possibility to switch back if it somehow wouldn't have turned out to be interesting. But I never was tempted because it really intrigued me so much to work on ants. Yeah.
So let's come back a little bit to your superorganism and to the complexity of the interactions. You recently published a paper on the abdominal microbiome of fungus growing or fungus farming ants. So it's clearly not only that on the skin or the cuticle of the ants, but also, of course, as in any other organism, there is no epithelium which is free of microbes. And now you have understood all the complex social interactions, or you're approaching them. Now, it's getting, obviously, with every 5 years or 10 years of research, you discover more previously invisible complexity. And that's, I think, due to our modern technologies. What's the role of the abdominal bacteria now? Is that helping digestion? Or what do you think are the abdominal bacteria doing in this tripartite symbiosis, as you want to say? We have the fungi, we have the ants, and now we have the bacteria. Well,
digestion is something very special in these ants, because if you switch, and this happened about 60 million years ago, to eating only fungus, because that's what they do. So not only do they rear their own food, this is all they eat. Your gut microbiome becomes special because that's easy digestible food. And so what would you need in your guts to facilitate that? And we became interested just to find out, because we didn't really have a clear idea what we would find, but meta sequencing came along and we decided to give it a shot and to sort of look at that throughout the phylogenetic tree, because we work in Panama where we get our ants and many of the branches are represented there, so you could do it in a comparative way. And the idea that we had was that maybe we shouldn't necessarily look for some form of optimal diversity, but really experiencing that we had so much specialization in these multi-layered symbioses that we might actually find some dominant bacteria that might be additional symbionts, that might be providing very specific services to this complex meta-organism, as you would call it. And in fact, that's what we found. So we find that when fungus farming started, so in the basal branches of the phylogeny, there's actually very, very few bacteria in these guts. They're diverse, but it's very hard to sort of pinpoint that you have very specific symbionts.
A few bacteria, you mean few phylotypes? Or there's one dominant one and then some minor ones?
Nothing's dominant there.
Okay.
You just have bits of a lot of different things. But as the symbiosis became more sophisticated by secondary developments in the scale of fungus farming, in the size of the colonies, we were also discovering that the bacterial abundances went up, so the titers went up, so there were just many more of them. We started to find them in specific organs associated with the guts, and we could clearly see there were just rather few main players there. And the bacteria we studied specifically because we could isolate them in genome sequencing were molecules. These are in many ways very interesting bacteria. They're very tiny, they don't have cell walls, and they're relatively not so much studied. But it turned out that actually there were several strains that were really sometimes abundant in the intestinal systems of these ants. So we were starting to
live extracellularly or intracellularly?
Well, it's a good question. It turns out both. Okay. So that this is my postdoc, Panos Sapountzis, who did all this work. He could clearly see when using fish microscopy, they were associated with fat body type of tissues. But we were also finding them in the hindgut. And so it turned out that when we were reconstructing their pathways through that genome sequencing thing, they were doing some very interesting things for the symbiosis that in retrospect made a lot of sense. So the main thing that we found they were doing was to recycle arginine. Now arginine we had already encountered as a very interesting amino acid in this symbiosis, because when a couple of years ago we published a comparative genomic paper, we could see that the ants had lost the ability to synthesize arginine. So very early on, so basically they outsourced that to the fungus garden.
A classical metabolic dependency.
Yes, and we believe that that must have been very instrumental in actually making the symbiosis obligate, because if you sort of outsource such an important function, you need that fungus around you all the time to get the nitrogen in particular also, because arginine is the most nitrogen-rich amino acid. But then we started to realize, after we found that these molecules were able to break down arginine, after some time we realized that that actually makes sense to do that, because if you've outsourced such a function, it means you cannot have it delivered to you at an averagely sufficiently level. No, you need to avoid that you get too little of it, because that would threaten your life. So what you get from a fungus garden is enough or an excess. And that excess can become a waste if you don't know what to do with it as a meta-organism. So what we found was that these molecules, bacterial strains, they actually break down arginine into ammonia again, just before the ants produce their fecal droplets to manure their fungus garden. So in essence, they turn a complex organic molecule that is not immediately useful as fertilizer for new garden growth
into a fertilizer. How wonderful is that?
And that is sort of was really an additional piece of the little puzzle that It's all so fine-tuned and co-adapted.
Yeah.
But here was something that potentially was leaky in terms of wasting resources. And we only realized that after we looked at the pathways in these bacterial strains and suddenly realized, yes, that makes, in fact, perfect sense. As soon as you had a fungus garden symbiosis like that, you created a niche for some bacterial symbionts that you could wait for to evolve that would solve this recycling problem for you.
That's a wonderful story. A number of people in the host microbiome, particularly in the intestinal microbiome field, are providing more and more evidence that among these, beside the metabolic interaction and so the digestion issues, microbial metabolites or products are somehow making it to the central nervous system, so the gut-brain axis, and so there's clear evidence that there's, at least in some systems, that central nervous function and output behavior is somehow influenced by microbial metabolites. Could you think, if you have not found it yet, but that's then a question in the future, but would it be possible that ants have a very complex behavior? Now you talk about a highly co-evolved, complex association of many different organisms. Could you think that that is the microbial symbionts and residents, for example, could they play a role in fine-tuning of ant behavior to support the superorganism?
It's a very good question. I'm afraid I have no answers to offer at this stage. It is clearly a next thing you would like to know, but it will also be a rather challenging thing to do, because you would have to do experiments sort of trying to manipulate, say, the composition. Can you make
germ-free ants?
No, that is, I think, not possible. We've tried to do bits and pieces of that in the published work, so you can use antibiotics to clear out at least most of the intestinal gut bacteria. But you also see you damage the ants, so it is not something that seems to be sustainable. And here we may be handicapped a little bit by that the microbiome is so co-adapted, if you like. So even though we don't think all these bacteria are needed all the time, these few specialized symbionts, but trying to knock them out may actually sort of mean you create dysfunctional ants. So this is where we're running into, I think, a handicap of not having a true model system that you can manipulate for 100%, which is almost possible in others. It's more difficult. So we would have to work with, say, experimentally manipulating the amount of bacteria, or looking at if you have ants with this strain rather than that strain, would that make a difference for how they express certain behaviors? But I would actually not be surprised if there were links there, because the entire system is so tightly integrated. And if somehow bacteria became a useful thing to have to tell you if you have a lot of these bacteria to solve a particular challenge in the symbiosis, maybe there is a set of behaviors that would be associated to that. So I wouldn't be at all surprised if we could find that. Having said that, I know this is a hot topic of research in honeybees at the moment. And maybe in the short run, that system is more likely to provide some answers, because there is a longer tradition of doing microbiome work in the honeybee than in the aptine ants. But it's certainly a valid question.
Looking a little bit in the future, and what you would, from your experienced point of view, where do you think, you mentioned already a few things, where do you think the ant sociobiology field will move or should move for the next 5 years from now?
Well, you can only make educated guesses, because developments go very fast these days. But I think we can be sure there will be huge big data elements in where we're going towards. This will not only be studies of gene expression, but in particular also mapping what bacteria are there, what they do. This is a field with huge potential, and the technology becomes more and more accessible, cheap, easy to handle. So I think I will keep an interest in these things and try to contribute to further development of the general role of microbiomes in ants, not only in the fungus growers, but across the board. We're also planning to sequence many more ant genomes, and we take the microbiomes along. Having said that, the other thing that increasingly, as I become more senior, interests me is to try to do the theoretical synthesis part, because I think the two should go together. There is a tendency these days for younger researchers to think that the big data is everything. That's what we need to become good at. My take is that that's important, but you will need social evolution theory to ask the right questions to your big data things. Otherwise, it becomes a fishing expedition, which is not necessarily very efficient. And sometimes when I think back, you can clearly see that the history of biology has gone through these phases where we either had only data, and then at some point we get theory. Like there was 100 years between Linnaeus and Darwin, and it was all description, and it was all monitoring. And then Darwin comes along and makes sense out of why we have what we have in nature. And I think we should try and avoid we go into a new purely data-driven phase for biology by actually making sure that social evolution theory is clearly outlined to predict that if we start looking at metagenomes of bacteria, there's certain things maybe that we expect in terms of adaptations, and other things we don't, because I'm firmly convinced that we'll make our big data research much more productive if it's guided by theory and we can make it hypothesis testing rather than just see what happens.
Yeah, a very important point. I think it is in these highly technologically driven times. Not forget the theory, and I think a person with such an experience is, of course, just the person to do that. Last question: if you could press the redo button, think back. You have a splendid career. You're recognized. Would you do anything different if you could start from the beginning? That's question A. And question B would be, if a young person comes to you and asks about advice how to make a successful career in academia, are there very few points which you may tell them?
All right.
Press the button and go back.
Start at the beginning. It's always interesting to reflect on that. I have no regrets. I wouldn't want to do anything differently if I look back on it. Having said that, having grown to the stage of a career I'm in, it's also very clear that it's very hard to predict when you're a young person where you will end up going.
Absolutely.
And that's particularly true, and this would be one of my main advices to young people. If you would really think it's central to just pursue your curiosity. Now, I realize that's easier said than done, and I've been fortunate in my career to sort of be in academic settings and have the grants to largely be able to pursue a career like that. But there's things that may help to actually keep doing that, and that one of them is to basically, if you want to pursue a career in academia, to very early on try to read extremely broadly and to somehow find a subfield, a question, where you can even explain to your grandma why it is interesting. Why it is novel. Because if you can do that, you're also more likely to somehow convince your senior colleagues that you may be worth some attention. And that's another, I think, aspect. Getting your own ideas, getting them as early as possible, and then be visible whenever you have a chance at conferences. And try to talk to the more senior people, because you maybe find that very frightening, you may be shy to do that, and they may be a bit hard to find and to actually persuade them to give you a little bit of time in such a discussion. But I think you should also realize that these people are really looking out for young talent. And if you have an interesting story to tell, the likelihood they'd be interested in listening is actually much higher than you might fear it to be. And personally, I've been extremely fortunate to have worked with many very talented young people that I started to chat with at conferences, and often made me develop new lines of research from a profound natural history interest. But always like, has nobody worked on this question before? Could you do that? Could we get funding to do that? And quite often I just initiated those lines. Parasitic butterfly larvae pestering ant colonies. We started an independent line on fungus growing termites to have a parallel system to the fungus growing ants. I initiated a program on zombie ants. And all these people that came to work with me took that program with them after having established. And I found it so intellectually stimulating to go to these sort of innovation process, because they were also innovative for me to help me to think broader and out of the box by not sticking necessarily to the same system, and to have these wonderful interactions with young people. But of course, I mean, if you're in that junior position, it is your drive that in the end should make it happen. And that remains central. And so keep steering your own ideas and your own imagination.
Yeah. Find your own question and stick to the question. Yeah. Thank you so much. I hope that many young people listen to you when they are little in turning points of their career. Thank you so much, Koos, for talking to us. Thank you.
Well, my pleasure. Thank you.
Thank you.